morning to all present here. I request all the participants present and our respected dignitaries to maintain silence for 2 minutes. We will have Odu Mani. A very good morning to all present here. Respected Principal Madam, Dr. Supriya Shidare, Convener and Coordinator for Westcoast ATAL FDP, IQAC Coordinator, Dr. Rajeshri Hirlekar, Co-Coordinator for Westcoast ATAL FDP, Dr. Anita Aire, Organizing Committee Coordinator for Westcoast ATAL FDP. Today's speaker, Dr. Mandar Kothule, Chairman and CEO, IQ Pharma Private Limited, Organizing Committee members and my dear participants. I, Dr. Aparna Palshetkar, Assistant Professor from Department of Pharmaceutics, on behalf of Vivekananda Education Society's College of Pharmacy, welcome you all on day 3 of offline AICTE sponsored ATAL 1 week online and 1 week offline faculty development program on research and innovation ecosystem in academics need of the hour. I request Principal Madam to felicitate and welcome Dr. Mandar Kothule with a sapling. I would like you all to take a note. Today's session was being taken by Dr. Kothare sir, sorry. But due to unavoidable personal reasons, he cannot make it for this session. So we have one more learned speaker with us today, Dr. Mandar Kothule. It is my pleasure to introduce today's speaker, Dr. Mandar Kothule. Sir has completed his B.Pharm from Pune College of Pharmacy, University of Pune, M.Pharm and PhD in Tech from Bombay College of Pharmacy, University of Mumbai, Sir is a certified patent agent and valuator in India. Sir completed Oxford's Advanced Management and Leadership Program, SAFE Business School, University of Oxford in 2012. Dr. Mandar Kodkule has about 25 years of experience in the pharmaceutical industry in the areas of computer-aided drug designing, chemical research and technology transfer, intellectual property, business development, corporate strategy and strategic alliances with companies such as Ranbaxy Laboratories Limited, Landmark Pharmaceuticals Limited, Purohit Limited and IQGenX Pharma Private Limited. Sir has worked with Minister of Trade and Commerce and Technical Advisor Professor Marshalkar on finalization of draft for Indian Patent Act 2005 and its amendments in 26. He has successfully bagged Maximum Granted Patents Award by Government of India and Farm Excel for Purohit for 6 consecutively and maintained a 100% success record in US litigations. Sir has achieved DEFRA's Dr. Radhakrishnan Gold Medal Award for Outstanding Individual Achievement and Distinguished Service in Special Education in Pharmaceuticals in December 2012. Dr. Mandar Kodkule has received IP Gems of India Award given by Questel Group, France in June 2018 in recognition of services rendered in field of IP in pharmaceuticals and healthcare. Top Pharma Leaders Award given by Conference Gorilla in June 2019. Most Influential Leader in Packaging Industry Award conferred by National Congress and awards for sustainable exports and packaging in November 2019. Then 101 Most Fabulous Global Healthcare Leaders Award conferred by Economic Times Now and World Health and Wellness Congress in February 2020. Outstanding Leadership Award conferred by Health 2.0 in recognition of contribution to the healthcare industry in March 2022. Dr. Mandar Kodkule is affiliated as Expert Committee Member on Pharmacy Course for RUSA, that is Rashtriya Uchchatar Shikshan Abhiyan, Maharashtra State, and a member for various governmental and non-governmental trade organizations such as FICCI, CII, Farm Excel, IPA, IDMA, and Continental Who's Who. Sir is being a resource person for conferences conducted by IQPC, IBC Asia, IDMA, UBM, CPHI, and various others on pharmaceutical R&D, patent law, Hatch-Waxman Act, and strategic planning. Dr. Mandar Kodkule has 5 international research publications, 115 patent applications, and 22 granted patents to his credit. Presently, Dr. Mandar Kodkule is a chairman and CEO of IQGenX Pharma Private Limited. So with this brief introduction, I request Dr. Mandar Kodkule to start with his presentation and share his expertise on research and development, innovations and patents with the audience. Over to you, sir.
Thanks to Dr. Supriya also for invite, and I could make it on time because she just informed me a couple of days back. I was there, and we had a very long meeting. She was 3 years senior to me, Dr. Supriya. 4 years senior to me. that's pointed out by Dr. Ishmael, because he really seems to be, and I know you are also here, because we all are from College of Pharmacy, so nice to see some more faces also over here. And I'm happy that I started my journey in this with these three ladies which are sitting out here. And a lot of help, association has helped me to shape up my career also being involved in pharmacy. So when we talk about, I believe I am audible, because I don't want to hide behind that. If I am not audible, just tell me, but otherwise I should be audible. So we will, today's agenda we will talk about what is R&D. Before R&D we will learn a little bit about knowledge, what is knowledge, and why we should have knowledge, how we can improve our knowledge which is required for R&D. And also we will look at innovations, what are innovations, what are the current innovations that are happening in the industry. And briefly we will talk about patents, because patents is something which everybody knows. I believe you have courses on patents now. Most of you have done the WIPO courses, and a lot of information is available on the internet about patents. So my question to you, let's start with this question. Just write down in your area of expertise, out of 100%, how much you know, how much you don't know. So let's say 100%, okay, you know how much you know in terms of percentage and how much you don't know. Okay. Who invented gravity? Isaac Newton. According to you, how much Newton knew about gravity? Percent-wise. Anybody?
In the appendix, 0%.
You have given some amount of credit. So the scientists believe that Newton knew 0.4% about gravity. Now look at your answers, what you have written about area that you know. So, because something we knew more about gravity from Einstein, then from Stephen Hawking, and so many others. So it's an emerging area. The moment we think that we know, we become confident and we don't learn. We don't get our knowledge. Okay. So having a fair assessment about what we know is very critical when you are an educator or a researcher. Okay. And why so? So there is a word called as ligometry. What do you mean by ligo? Ligo in Latin means shadow. Something which is not there in light. Something which, in other words, we don't know. It's in shadows. We don't know about it. Exists, but not seen. Metry, means measurement. So ligometry means quantifying what we don't know. And for that, we need to know what we don't know. And I told you why it is important. Because if you know what you don't know, you will make an effort to know it. So this is ligometry. This boundary, what we are looking at. This boundary. And then there is a vast area which we don't know. And when your goal is to gain knowledge, failure is no longer possible. You have to gain knowledge. You have to understand basically that you have to first admit the fact that I don't know. And I can tell you from my experience, how many of you been to Louvre Museum in Paris? Anybody seen Louvre Museum? Yeah. Room number 7, South Wing. Okay. That room number 7 has a wall in between, so there is a north entrance, there is a south entrance. Unfortunately, I entered from the south entrance. I was looking at, and there is a wall. I was looking at 5 beautiful pictures. Beautiful. There was a Van Gogh, there was a Ravindra. 5 beautiful pictures. Unfortunately, on that south side, I was the only person. Only. Everybody was just walking past all those beautiful paintings of Ravindra, Van Gogh, and others, and they were going towards the north side. So, out of curiosity, I went down to the north side. 400 people at least, 400 on the south side, no space to stand. What was there? Da Vinci's Mona Lisa. And if you look at that, you would realize what a beauty, what a masterpiece it is. Okay? So where the value comes from? If you look at those other 5 pictures, which are on the paintings on the other side of the wall, not even 20 feet apart, nobody is looking at them. Everybody is looking at Mona Lisa. So what we have to, and Van Gogh spent at least the same amount of time which probably Da Vinci took to paint those pictures. Okay. So where you have to spend your time in creating value, where 400 people are interested in looking at your art, looking at what you are doing, looking at what you are doing as a research, rather than just a masterpiece, good for nothing, and nobody wants to see. That's why it is very much necessary to quantify what you don't know. What we know. So the best, fairest assumption is I don't know anything, let me start by knowing something. So you will make an effort. If you make an effort, you will be creative and you won't do any faulty planning. This is the basics of liability. Now why, as an educator or as a student or as a researcher, you should know about liability? Throughout our up to BPharm, right from AG, senior AG, whatever, Montessori, first, second standard, 10, 12, BPharm, you've been told about what others know. Has any teacher, has any professor or anybody told you that in pharmaceutics or in biotechnology or in dispensing, we don't know this. Has anybody made you curious to think that, look, we don't know this. How this happens, we don't know. There are always some solution, there is some explanation to the Bayes rule or why amine reacts in such a way with an alcohol or how amide is formed. There is explanation to that. Problems, we are not good. We are not taught. So till B Farm, we are told what the world knows. After that, when we come to N Farm, it's the first time we start looking at the problem. That there is something could be a problem. And once we know that something could be a problem, we start looking at how we are going to address that problem. We get only 3 semesters, 18 months, of which roughly about 12 months get into writing papers. Okay. So we hardly get any time to think about that problem and solve it. In that also you are expected to write your thesis, right? I don't know, now you are supposed to write literature, article also, patent also, or so many things you have to do. And to find a problem also is very difficult, right? As an educator, if you are depending on the problems to be identified, you are looking at resources, how many resources are available to me, whether the drug is available, what kind of problem I am going to give to my student. Because the student also wants fast work to be done. It's not just the educator's problem. But in this process, what happens is you start at least getting into an area where you are doing research and not something which is told to you as a part of you. Okay. There are unknowns. So you are chartering into unknown area, and that's the only difference between BPharm, M.Pharm and PhD. Nothing else. Don't think that after completing BPharm, after completing M.Pharm or PhD, you become master in the subject. No. BPharm person with 10 years of experience, probably, or six years of experience, probably is of the same capability. What you have done differently is you have started doing something called as problem solution approach. And how knowledge is related to the failure. So, if you look at knowledge management, which every institute does that, knowledge management, how much knowledge is being generated, every institute, every corporation does this. So, in 2014, Harvard came up with a study and they said that 100% of all the problems in a corporation are related to knowledge management, not managing the knowledge problem. Also, this is 20%. I know when I don't know. Assuming that I know, that's a 20% failure. I know how to do it. Rather than doing it the right way, people do it with old mindset, not looking at why people failed, other people failed doing it. We underestimate competition when we try and gain our own knowledge. We underestimate that somebody else will not come up with a completely different solution. Look at this way. 1890, two brothers in US, Sullivan brothers, used to go to the lakes in the winter season, cut the ice from there, get into the storage facility, store the ice, and then whoever comes, distribute the ice to them. So that's how ice harvesting started. Right? That was ice 1.0. 2.0, this ice factory was built. And from there the ice was cut and distributed to those who need it. What is ice 3.0? You know? Fridge that we have at our home. We don't need to go to the ice factory. That's disruption because it disrupted the industry, the huge facilities that were created to prepare and store ice. Kodak, if you remember, didn't believe in something called as storage. They still believe that the films are the best way. They didn't believe on even memory storage can store your photographs beautifully. Nobody needs prints now. I don't think anybody takes prints. How many of you take prints of photographs? Nobody. It's there. But we take lots and lots of photographs every day. Right? It was not possible 20 years back, by the way. Those of you know that 20 years back, the only way possible to have a photograph is by print. So that's disruption, complete disruption in the technology. So we underestimate the competition and don't create new knowledge. Managing knowledge, 40% of the people. So what all this comes out to be, you will fail. If you're trying to do research, you will fail. 99, 100%. Because that's research, that's black and white. It's not black and white, it's grey. You have to, there is unknowns in that. And in managing unknowns, you will fail. But you will learn. So if you don't fail, you will not learn anything. So fail. No problem. But if you repeat your failures, you're the stupidest person on. Because if you make the same mistake twice, then it's not a mistake, it's your choice. Knowing that somebody is already falling, you're still walking on the same path and falling into the ditch. This is by Einstein. This insanity, doing the same thing over and over again and expecting different results. So there is a theory about research. So now what is research? What is research? Plain simple terms. You know that there is something prior knowledge and prior research. If somebody has done some work, there is a method available for HPLC determination of a particular drug. Maybe it is there in USP, maybe there is some article. And you are trying, you tried that, faced a problem and you said, yes, there is a problem. So I identified a problem in the prior knowledge and you came up with a hypothesis that if I used, instead of methanol, acetonitrile, or if I use this particular column or I change the flow rate or I do something complexing, some kind of muting, it will give me a better result. So you have a hypothesis. Based on that hypothesis, you design few experiments. You run those experiments. You got the results. You analyze the data, came to a conclusion that whether it is working in the right way or not, and then you thought a way forward. How am I going to do it better? Again, you are comparing. So it's not a one-way process, it's a cycle. If you are a true researcher, you will not stop at any particular point because data will give you some, point out to you to some other problems. Right? Because solution to a problem is a problem. The best solution is which leads you to the lowest problem. Okay. So, for example, problem is you are not a profitable company. You made a loss of, let's say, 10 crores. Okay. In a particular year, you made a loss of 10 crores. Now, what is the best thing to happen? You made 100 crores profit, right? But if in next year you make a loss of 7 crores, you're good, right? Because from 10 crores of loss, you at least save 3 crores of loss. If you can't get into profitability, at least you have to stop loss and you have to think. So that's, if your hypothesis, I'm not going to make 10 crores of loss, I'm going to make, let's say, 1 crore of profit. Halfway down the year, you realize that's not possible. What do you do? You change your plan, you will change your assumptions, you will change your design completely. It's also a research not necessary. So anything which involves a problem solution approach is a research. So what in that you are doing, ideally you should be doing is you should be, when you are into research, you should be looking at what is the unmet need. Is there an unmet need? What is unmet need is some problem which is not addressed by any other person. Is there an unmet need? Now, do I have resources? If I identify unmet need, the next thing is, do I have the resources? Do I have the drugs? Do I know from somewhere I can get those drugs on which I want to do some work? Maybe analytical work, maybe formulation work, or whatever that is. Do I have impurities? Do I have the working standard? From where I am going to get it? Solvents, materials, excipients, packaging material, whatever that is required, stability, XRD, NMR, all the analytical tools. If I don't have in my lab or in college, where am I going to get it? So those resources in terms of materials, money, time and personnel, who am I going to research is not an individual activity. Research is a teamwork. So when I was doing my M.Pharm and PhD in pharmacology and pharmacy, I relied very heavily on the pharmacology team. Fortunately, we had a wonderful pharmacology team in pharmacology and pharmacy. They did all the experiments which were there to be done on my compounds. They helped me doing it. So, and I learned so many things by doing that, but I am not an expert in putting or taking out a heart and mounting it. That's not something which is my fault. And I don't require to do also that, right? Because I was not going to do heart dissections after I completed my PhD. I went into a completely different area. So as long as I know and I took their help, I was fine. So you need to understand also which are the personnel you are going to take help from. If you want to fail, or if you are going to fail, fail early and fail cheap. Because if you fail at a later point in time, the cost is too high and you lost a lot of time. So whatever stupid ideas that you have in your mind, start with them first. That gives you a lot of spread in terms of what works and what doesn't work. In our industry also, what we do, something which is called as quality by design. So quality by design will also tell you exactly the same thing. You should know the lowest and the highest limit in which your product is going to work and your method is going to work. Whether the coating, extended release or functional coating has to be 4%, then you should know what happens if you do 1% and you should know what happens if there is a 7% coating. These are the worst case situations, right? But knowing this, 2 points, doing it early will give you a lot of inputs. What happens if you hit in the first trial itself, you get the result, you are very happy and you start reproducing that. You have not learned anything about the product. You have to fail. You should know how it will not work rather than how it works. Okay, so if you want to do that early enough in your research time. So fail early, fail cheap. First time right. So once you have everything, okay, once you failed, once you got a prototype in your hand, make an effort to do it first time right. Don't fail after that. The first experiment that you do, after that reproducibility, scale up, whatever that is, that has to be absolutely perfect. Understand why you are doing this. Get to the basics. Before doing, you should know why you are doing that, what you are going to do, how you are going to do it. Most importantly, unless it has scale up, unless you understand the scale up issues, don't start with any invention. At institute level, we think a lot about, nanoparticulate technologies, liposomal technologies, or various different things. Good, fantastic. How many products actually exist in market on those technologies? And in terms of learning, if that is giving you something good, but you should understand the commercial aspects also. How am I going to commercialize the research that I am going to do? Is there a way I can think of some way of commercializing, scaling up? So innovations. Steve Jobs said that innovation distinguishes between a leader and a follower. What is innovation, by the way? Anybody has any idea? What is innovation?
When an idea is commercialized, when something novel is
something which is not known. Okay. So there are different types of innovations. We will look at it. But innovation is basically means something which is not known. You're doing it for the first time. Got the result, positive result. It is applicable. It has some So three things: novelty, inventive step, that's non-obviousness. Inventive step is non-obviousness, and industrial application. That's it. Innovation. Now, why Steve Jobs? Why Apple is called as an innovative company? Look at it. How disruption was done, continuous disruption. So Mac in 1984, 2001 iPod, I don't think nobody uses iPod any these days, but that time iPod, how many of you had an iPod? One of the best tools, right? iPod mini, you used to have 800 songs on one drive. Those were the era of audio cassettes and CDs that time. Then iPhone, MacBook, iPad, so on and so forth. Now it is the 14. Just a couple of days back we launched iPhone 14, Mac. I don't know which version is currently going on, but they are the leaders. So now compare it with the pharma industry. Are we doing disruptive innovations? Yes, in pharma also. Look at diabetes treatment, how it evolved. So he started with metformin way back 60s. In 70s, realized that these bovine and porcine insulins can be used. So bovine porcine insulins went to sulfonylureas, glyburide, galantamide, glimepiride, glycoside, all those drugs. Then insulin, recombinant insulin, human insulin, from essentially it was initially from yeast, then bacteria, then mammalian cell. So recombinant technology also improved. Then insulin analog stage, short, medium, long acting, aspartame, Largy, all those. They came into picture. Okay. Does it stop over there? No, it has not. Then came the pioglitazone, rosiglitazone, glitazones. Then GLP-1 analogs, and these are still continuing. Exenatide, liraglutide. Now you have oral GLP analogs, semaglutide. Once a day. And there is one more glutide which is there in the market, which is once in 14 days. Orally, once in 14 days. So you can look at it, how the technology is improving. As we understand more about the disease, then DPP-4 inhibitors, sitagliptin, saxagliptin, sita, and then you have frozen SGLT4 inhibitors, canagliflozin, dapagliflozin, empagliflozin, and it is still going on. Now who knows? Tomorrow some monoclonal antibody will be available for treatment of diabetes. Okay. So this is how the innovation never ends. It never stops in a particular disease. If you look at cardiac, you started with identifying first epinephrine or adrenaline as autopoid, which is present in the body. Then you came up with agonist, antagonist, all those beta, alpha receptor, and the whole science improved. Then it just didn't stop over there. Then you got all those depends, calcium channel blockers, Ravindra, FDP, all those. Then came ACE inhibitors, then so many and so forth. So all these research, sartans, all the way up to sartans, US sartans. So basically, what is strategy? Generic question. What is strategy? Plan? Planning of something is strategy. Okay? What is strategy? Planning and methodology. Okay. outcome-based. Outcome-based planning. Okay. If I tell you planning is one part of strategy, it's not strategy. What is strategy? No, strategy is simply art of creating superior performance. That's it. So better than last time. I told you, don't lose. If you are losing 10 crores last year, superior performance is even losing 9.8 crores. But you can have that as a strategy, right? So strategy is creating year on year, you have to be better. So ideally, you require positioning in strategy. That Apple has positioned themselves as, I'm not worried at what cost people buy it, but I'm going to give a very innovative product, maybe the cost is very high. On the other hand, you have all these Chinese phones available, commodity items, cheap availability, okay, 2,000, 1,500, 1,000 rupees, 800 rupees, phones are available, right? Those phones are not innovative phones. They are just, phones. Because you just need what in a phone you need. Talk, send messages, maybe WhatsApp, maybe Twitter, maybe Facebook, social media. That's it. That's all you need. And those phones give that. Okay. So positioning of Apple is different, positioning of Huawei is different. Different positioning. Which is good, which is bad, let's not talk about it. It's eventually the performance that matters. Okay. Avoid being best. Because if you are best, tomorrow somebody else will be. So what you need to look at when you undergoing a research strategy, when you're making an innovation strategy, you've got to be unique. So you're way ahead of competition. I have to be unique. I need not be best. But I got to be unique. Because when I'm unique, somebody will follow me. Or I'm creating something which is niche in the area where nobody is already there. So there is a value for my research. And it's sustainable over a long period, not just 1 or 2 years. I need to sustain over a long period. When strategy is improved or superior performance. And that superior performance has to last over a period. So there is a short-term strategy, there is a mid-term strategy, there is a long-term strategy. All different. And your goals, your objectives will be different for all these three things. When you have a short-term strategy, mid-term strategy, long-term strategy, your goals, your resources, your planning will be different. Is this visible? So, how pharma innovations, what was the trend so far in pharma innovations? The trend is first in early 60s, 50s and 60s, how we can reduce the mortality. Because there were infections, there were diseases, which led to huge mortality rates. So how we are going to reduce the mortality, that was the first innovation challenges. So all these, antibiotics, vaccines, all these games started coming in those years. How to treat fever, if you look at it. Lifestyle diseases came much later. The metabolic, diabetes, blood pressure, all these came much later in terms of research in late 80s and early 90s. Because the problem of mortality was addressed in the late 80s and early 90s. So people started looking at how I can improve the life expectancy. So once mortality is stopped, how the life can be improved or extended. Average at that point in time globally was about 55 years. In certain countries it was much higher, but in most of the developing countries or underdeveloped countries, the life expectancy was much lower. So average was 55. So scientists took a World Health Organization, WHO, took a target that it has to be at least 70 or 65, which they were able to achieve partially in 2000. That changed again the innovation trend. Fewer side effects. So once you have targeted mortality, reduced mortality, improved life expectancy, how can I reduce the side effects of the existing drugs? So that's in 2000, you would see improved tolerability and fewer side effects came into picture. And those are the guidelines also which got into regulators and ICH and all those things. Then came faster recovery. Faster recovery. How can I get the recovery in the fastest possible way? I don't want to waste time because time was critical over here in 2005, somewhere around 2005. Faster recovery, and last it came to quality of life. So now the era is how I can improve the quality of life. So nutraceuticals and all those preventive rather than treatment, preventive medicines are coming into picture now. Quality of life also means the delivery of the drug. How am I delivering the drug? So devices, packaging, aesthetic also plays a very important role. Now look at these numbers. Okay. This is across the industry. This is not just pharma. Pharma spends roughly about 70-80 billion dollars year on year in innovation. Way higher than you take any industry, technology, automobiles, aerospace, defense, telecom, healthcare equipment, oil and gas. So pharma spends more on innovation even compared to defense. Can you believe that? That much money is available in pharma and it is growing by 5% year on year, 2008 to 9. In COVID times, it was exponentially high. Okay. Now since people are more aware about biological warfare, chemical warfare and all these things, and people are also aware that with this lockdown, how much mentally and physically people health or general people health is also affected. There is a lot of trauma, there is a lot of anxiety, there is a lot of depression, obesity, heart diseases, because almost 2 years people did nothing. So there are a lot of issues and there is a lot of spend on that. So these numbers are skewed up even further. So pharma spends very high. These are some of the numbers. So over about 100 billion US dollars are spent on pharma research, and 10 leading global R&D firms, 5 R&D firms out of 10 global companies are pharma. That much spend is there. Now, what are the challenges in the innovation? So, if you look at rising cost, it's one of the biggest problems. Cost is going up. Pharma innovation cost is going up. Why? Because the regulations are getting stricter. The materials are not available so easily. The targets, the clinical trials that are involved, the competition that is there has gone up. So everywhere, if you want to create something, you are spending more. You are also regulatory and compliance has gone up. Everything now needs to be documented. Everything needs to be done in the QB way, the GLP way, the GCP way. All those regulations are brought into picture. Innovation capacity, the pool. Unfortunately, what has happened over the last decade is that the AI that has come up or the rational drug discovery that has come up has kind of killed the innovation capability of a younger generation. Google is available. Solution. That's a solution, right? Google is a solution now for everything. Rather than spending time in library and thinking about the problem, we put that problem across onto the Facebook, Twitter, all those things. We get 20 suggestions. 19 of them are useless. And the one impractical remaining. Okay? So, we are from a Google University. We stopped using the technical university. The thinking capability has gone down significantly over the last 10 years. I don't know whether you have observed it or not. We think we know everything. So coming back to ligometry, we don't improve our knowledge because our knowledge is Google. That's a basic problem. So our innovation capacity globally has gone down at least by 33%. This is the estimate by World Bank. Tough patent law. So patent laws, because of the ongoing cases that are happening, are becoming stricter and stricter. So if you go for a new innovation, not necessarily you will get the rights for that particular invention in terms of patent. You will get not basically a good protection. So the time amount spent versus the protection that you got is discouraging, so you are not doing any research. Okay. Also funding. So 110 billion fund is available, but to whom it is available? Not to me. Right? So how am I going to get? How am I going to resolve my funding issues? Am I going to approach the VCs, individual investors or corporates or whom? Or the government agencies to fund me? How am I going to write those funding proposals? Am I going to get those funds? So funding issues are still there, and expectations, rising expectations. So these are some of the reasons why pharma innovation has become time consuming and very high rate of failures. So assumptions are not being properly made because technology is evolving so much. Now if you look at COVID time, there were 79 vaccines which were studied. 79. Eventually, how many were qualified? 4. Only 4. Pfizer, Moderna, Johnson & Johnson, Bharat Serum Institute, Russia. Chinese vaccine is not approved from anywhere except China. Okay, so many companies, only 5 vaccines. Everybody was working, spending billions of dollars. How many drugs that doctor tried? Remdesivir, molnupiravir, favipiravir, tocilizumab, you name it, all these drugs, none were successful. Okay, so spending, you look at repurposing, how much repurposing happened. Favipiravir was not supposed to be used for COVID. It was supposed to be used for influenza. But they repurposed the drug somehow, thought that this will work better. Remdesivir was supposed to be an influenza drug. But they tried it, it worked. It killed more patients than to cure. Eventually, Remdesivir was responsible for killing more COVID patients because of the negligence of doctors, not because of Remdesivir itself. 6 treatment doses supposed to be given, doctors gave 12, 15, 18 doses of Remdesivir, liver failure. That was it. So, if you look at really in pharma, innovation is much more tougher now. So, defining a problem, searching a problem is also very critical. And the result is, drying of pipeline. So, if you look at how many new drugs approved over the last 5 years, there is a 80% drop compared to, if you look at between 2000 and 2010, there were approximately about 790 new drugs. 790. So yearly about 80 new drugs were launched. Compare that with 2010 to 20, only 88 new drugs were launched. So drying pipeline, right? Nobody wants to invest. And everybody, all those drugs, the real only, if you look at new inventions, were only about 55, so 50%. The 50% were incremental innovations. And we will talk about it after some time. And Me Too strategies. What is that? That also we will talk about. So what is happening now in innovation in pharma? If you look at NCE or new chemical entity, people are looking at molecular targets rather than the same molecular target. Within that, there are enough dipins, there are enough sartans, there are enough prils, there are enough litazones, clozins, gliptins, there are enough of those statins. So people are now looking at new molecular targets where a new, conceptually new disease targeting can be done. Treatment can be involved. Improving profiles of existing drugs, repurposing, drug repurposing also, and that is new use of discarded products, but also very importantly improving profiles of existing drugs. Now, if you look at in oncologicals especially, you will find lot of profile improvement is happening through some kind of conjugation or complexation and then reducing the toxicity of the drug. Okay. Repurposing, I think the entire COVID theory was COVID treatment came out of repurpose. So discarded products, nobody was using them, just they tried to use it for COVID medicines and something worked. Even in Ayurveda, the same thing happened. Whatever the new medicines for COVID treatment were available, were all essentially used for cough, cold, sneezing, and TB medicines. So they were targeted towards that virus. Some of them work, some of them partially work, but that's the theory which is coming up. combinations of existing drugs. So the synergistic effect, reduced dose, this is tried at a very high level. I mean, this is at a very advanced level, that if I can reduce the dose of one of the drug which causes side effect by giving some other drug, okay, and that's where combination of NSAIDs with proton pump inhibitors and others came into existence. There is a one product available in the US market, which is the combination of dextromethorphan with quinidine. Now, dextromethorphan, we know it's a cough and cold medicines. Okay. A normal dose of dextromethorphan is about 40 mg. This is the drug which is used for pseudobulbar. What is pseudobulbar effect? It's sudden episodes of laughing and crying. Sudden episodes. So basically something in the CNS. US, all crazies, all type of disease you will find in US, which normally in India you won't find, because we don't have ADHD problems so much. Attention deficit hyperactivity disorder. You know how much is the market for ADHD drugs in the US? 16 billion. Which are the drugs? Methylphenidate, amphetamines, all narcotic products. Who are consuming? Kids between the age of 4 to 18. ADHD. That's ADHD. Okay. Pain therapy, which is the highest selling medicine in US, in terms of volume. Hydrocodone plus paracetamol. In US they call it ATAL. Acetaminophen. Okay. So Vicodin is the brand name. 7 billion units. How many? 7 billion units. What is the population of US? Roughly about 330 million. 40 tablets per person in US a year. I don't know. So, dextromethorphan is used in 20 milligram. How come they could come to 20 milligrams? Because they use quinidine, which actually is a blocker of cytochrome P450, which metabolizes dextromethorphan. Okay, so these are some of the strategies that are used. It's not just whether they are working synergistically or not. If something is blocking, like levodopa and carbidopa, ideally combination. Okay. Levodopa is a drug. Carbidopa is something which blocks the dopa reductase enzyme. Right? And you put entacapone into that. Entacapone is a COMT inhibitor, which also is responsible for dopa breakdown. So you have a drug. So combination. How do you prove the efficacy of one particular drug by using two different drugs in the combination? Improving care. So post-operative care, any operation, the post-operative care is normally pain management and disease or infection management. So in that there are combinations available. So how you can do a post-operative care or pre-operative care. So pre-operative medicines also, where during the operation you need to block a lot of your muscarinic responses. So normally the drugs like neostigmine or all of them are used. Now you have to block. Once you block, you have to reverse that effect also, right? After the management. after the operation is over, you have to reverse the effect also. So there is propuronium and all these, So Sugamadex has come, which is a new product. So Sugamadex in combination now is being used with cisatracurium and Sugamadex. In ophthalmics, you will find that how you can take improvement care. A lot of different examples. Altering side effects. So, side effect of one is altered, like combination of a NSAID with a PPI or a H2 receptor antagonist. Patient compliance. This is almost 90% of new innovations are happening in that area. Patient compliance. Okay. Dosing frequency. How can I make a drug once a day rather than twice a day or thrice a day? So, NDDs kind of new drug delivery systems, extended release, modified release, chronally released, what was that? And improving abuse potential. So certain drugs are, abused, like morphine and other. So there is a anti-abuse medicines that are available. Improve delivery devices. How can I improve the delivery devices? So earlier we were using vials for injection. So the doctor or the nurse used to take something out of it and then dilute it or inject that through a syringe. Now, to avoid that, pre-filled syringes came, okay, which are directly you can use. Pre-filled syringe, no need for a nurse or doctor. Now, as a pharmacist, we don't understand the impact of that. But the real impact comes when some medicine is available in vial, you are actually going to the hospital to administer it. So that cost, you have to pay to the hospital, the nursing staff cost, the syringe cost, the needle cost, everything is going to be thrown anyways. Okay, so you are adding so many costs. When you are giving something in PFS, prefilled syringe, all that cost can be avoided. The patient can be just trained how to take the PFS once or twice, and the patient can look at this from a insulin management perspective. So from PFS came auto injectors, pens, self-administration, EpiPen, insulin pens, one of the best examples. So patient doesn't have to go to the hospital to take the medicine. He just buys the pen, puts it, activate, take the dose on a daily basis. Very simple. Okay. Then came the bags, polybags, infusion bags, because the vial, you take the material, put it in a bag which contains the diluent and so many other things. So you are injecting it into a vial, something which is there into the vial, into the bag. If the dose is high, you need to take 20 ampules or 10 vials. Rather than that, there is something which is readily available in a bag, poly bag. So you just put the cannula onto the bag and start administering to the patient. That's a bag. Okay. Then there is an infusion pump. So there is a pump. It's not just the bag. To the bag attached is a pump where you can adjust the speed. The patient can drive while doing that. He need not be in the hospital. He need not be at home. He can do his work sitting on a computer. If the medication is not supposed to cause a drowsiness or something, just simple antibiotic or pain management therapy. Okay. So there are a lot of improvements that are happening on the delivery devices. If you look at even these compliance packs which are available in US, now in India also they have come up, wherein what we need to do is, if you are a diabetic person and normally a diabetes therapy or blood pressure management therapy requires taking lot many medicines, 7, 8 different drugs. So you need a statin, you need a beta blocker, or a calcium channel blocker, or a sartan, and you are taking along with that, let's say metformin, or CWP, or something. So you are taking actually 7, 8 medicines at a time. And you tend to forget because age wise. So what these compliance packs do is they put a day, okay, and all everything in one pack. The packet will do so that you know that all these medicines I have to take it on that day. If I forget, I know that this was supposed to be taken yesterday, I am not taking that. And you can talk to your doctor, or doctor can remind you. Now, there are, if you actually work on these, if somebody is a patient of asthma or pulmonary diseases and you are using those inhalers, okay, the inhaler sends you the reminder on your phone that you need to take your medicine now. A lot of technology is built into all these devices, auto-injector. They inform the patient that you need to take your dose of insulin now. Dose is delivered, whatever that is. So the patient also knows that if I am taking the dose, I don't know right earlier whether the dose is delivered or not. But now I know because the device says there is audio communication. The device tells me what to do. If I am using it for the first time, the device will tell me open the red cap. So I opened it. Once he knows that the patient has opened it, it talks about the next step to be done. So there are a lot of instructions are also built in on the device. So device things are improving. Personalized medicines. What is personalized medicine? There are a lot of dispensaries now in the US. Okay. So if you are allergic to something and you know that what works for you, you will get exactly that as a personalized medicine. Okay. And how they are making it? By 3D printing. Mostly by 3D printing. It's a compounding in 3D printing. The 3D printed tablet is available in the market. How many of you know? It's commercially available. Levetiracetam tablet in US is available, which is 3D printed. And normally, we in our lab are having a 3D printer. Wonderful. Very useful when you are looking at the HCP compatibility. Big studies. So I told you about, Taylorly failed with paint sheet. These are some of the tools, 3D printers for tablet compression. I mean, you don't need to compress a tablet. You don't need anything. You just need binder solution and powder, blend. That's it. So there are a lot of advantages when you are looking at patient compliance. Natural products. The basic thing that is happening is tissue culture. So everybody has looked at how to produce insulin from a plant, or how to produce vasopressin from a plant. Can I do some kind of tissue culturing, which I used to do it in bacteria and mammalian cells and yeast. I used to do that combination. Can I do it in plant? Because maintaining a plant is much simpler than maintaining a bacterial culture. Isolation is much easier when I am looking at plants than I am looking at bacteria. Okay. Most of the work is happening in natural products in terms of setting up what is the quality assurance and regulatory compliance criteria for natural products. A lot of work is happening on that. How to make it much more like regular pharmaceutical product, and the delivery, how I can rationalize the delivery. There are a lot of issues involved in elixirs and kadas, personal and tablet and all those things, but how I can rationalize it much better so that scale up and commercialization issues are resolved. In terms of diseases, these are by and large becoming more mature therapies. CDS, anti-infective, CNS, pain management, and acid reflux. These are becoming very mature therapies. No new inventions are happening over there. Where are the new inventions happening? Emerging therapies. All these. Anti-diabetics, endocrinology, oncology, antiviral, HIV, Hep C, now add to that COVID, add to that influenza, add to that the host of new biological warfare that is going to come up, and monoclonal antibodies, immunological diseases. So basically, these are the emerging therapies now. So in NCs, again, when we are talking about, there are new targets that are being looked at. We looked at that. Controlling the release of the drug. That is also the compliance, improving the compliance, how I can control the release. So once I am fully aware, normally what you would see is when a company comes up with an NC, new NC, their objective is to put that in the market. First put that in the market. So all the processes that are developed, the formulation that is developed is absolutely substantive. Because their objective is to get the patent life, which is more critical. Get the product in the market and then let's see, we will improve the formulation. So these are normally, the target candidates. If you look at what is there in phase 3 or phase 2, start looking at those drugs. Because they are the ones who are going to be launched 10 years down the line. Promising phase 3 candidate, it's something which you should look at as a target for new discovery. For doing something new in analytical, in process chemistry, in formulation. Because 90% it's an unexplored area by even the innovators. When you look at dosage form, the more focus, as I said, is on new delivery systems. So somehow now osmotic technology is picking up interest. Somehow this liposomal drug delivery system only for oncologicals is picking up. Somehow this technologies which are matrix kind of technologies, geomatrix technology, gastro-retentive technology is coming up. Reservoir technology is taking a lot of this thing because you can alter the release. The use of capsule surprisingly is improving a lot. Why? Simple reasons for capsule. The biggest advantage in a capsule is you don't need a compression machine. The moment you are out of that compression and coating machine, 90% of your problems in terms of showing scale up and commercialization are resolved. When you are in a capsule you can fill 4 different types of legs. You can fill granules, you can fill pellets, you can fill mini tablets, and a combination of all these. You can put immediate release pellets, modified release pellets, delayed release pellets, and a combination of them. So you can put a IR1, IR pellet,
ER1, ER2, ER. 4 different pellets in a capsule. The only disadvantage of a capsule is that it can fill only up to 500 or 550 milligrams. But now there are stress fit capsules available. So whatever it is, even you can put 1.2 grams of material in a stress fit capsule. It's called stress fit. Okay. So there are a lot of new things that are happening in capsule, and that's where it is becoming now. If you see, the ratio is completely towards tablet, and everybody prefers a tablet. But trends are changing with the new capsule material. Now that capsule is available in HPMC, which is called as a Beijing capsule. Now there will be Indian capsules available. There will be osmotic capsules. It's just the material. So change the material, you get whatever you want. Okay. Transdermal. In transdermal, what is more importantly happening is these something called as microneedles. Yeah. So microneedles is coming up in a big way, and those are biodegradable microneedles are also taking quite. So and what is more important in a dosage form, if you see, it's not the drug. As a pharmacist, we learn only about the drugs. We don't learn about the excipients. We don't learn about the packaging materials. We don't learn about the device. And what we have done in IQ is what we, right from day one, we signed a MOU with BASF, with Colorcon, who are the material suppliers. Short, which is for injectables, we supply packaging materials. Okay. We also signed up with ACG is an investor in our company. So they make the machines, they make the films and foils, they make the capsules. Now we understand 360 of the dosage form. So that's where you have to now put dose reduction, circadian, everything will happen if you have the right excipient, the right material. Because API comes with all these problems, but what is resolving the issue is the excipient, is the packaging, is the device that you are going to come up with. Device. So compliance kits, we talked about auto injectors, we talked about dry powder inhalers, meter dose inhalers. Now they tell you how to operate, how to do it, doses will work, everything. And it has become mandatory now. So if you're using a DPI in US, it is mandatory that the device should tell you the doses will work. At least some indication, visual or audio indication. Child resistant packs, painless injections. Now what is painless injection? Doesn't use needle. So how it gets administered? So what is used in that, ammonium nitrate, nitrous oxide, basically. So these are bombs. Actually, these are bombs, small bombs. So they create that much amount of pressure, so much of pressure that the fluid actually passes and crosses through your skin and dermis and enters into this systemic circulation. Maybe not. These are not essentially to be used as an IV kind of things, but these are normally either it will be intramuscular or mostly subcutaneous. Because with that kind of a force, if it penetrates the artery or the vein that is being used, artery normally, then help you out. what will happen, we don't know. So these are mostly subcutaneous. A lot of question mark again on the storage, on the use, on the abuse of these by the others, because these are banned materials as of now. If somebody can isolate those, it's good enough. Maybe 10 injections are good enough to grow this entire body protein. So, interesting area, but will it see the day of light? I'm not sure. Apart from that, these are the regular lab area, but all these are non-lab. Non-lab areas, regulated. The amount of innovation that is happening in the regulated area, because guidelines are changing. The requirements are changing, and the way these regulatory people are telling how driving the research, what to do, what not to do, what to be put, what kind of queries are anticipated, what kind of commercialization issues you will face. So all these things, quality, very important, because who is reviewing the documents, who is reviewing everything, who is managing the systems, is quality. Okay, so compliance, pharmacovigilance, IP, QBD, project management, technology management, there is something called as all the teams now, all the companies, big companies now have the technology management. Okay. And find out about more about that. Let me not tell you what is technology management is. Critical, crucial things. Logistic, supply chain, all these. Strategic planning, intelligence tracking. You should know what others are doing. So, a lot of new tools have come up in terms of intelligence tracking. New databases are available, but you have to continuously innovate in these areas also. Find out new ways of doing it, stay ahead of competition. So, we talked about innovation categories, right at the beginning. Why I'm telling you at the end is because two basic categories. Incrementally innovation. This is the space where 99% of innovations become. That's the space. Incremental innovation. Minor improvement, it could be from 1% to 10%, 12%, 15%. Improvement. No rocket science, that's where 99% of patents are filed. Incremental innovation. Then comes me too innovation. Somebody has invented propranolol, then somebody invented atenolol, then all those logs, somebody came up with nifedipine, then started the whole series of dipines, somebody came up with valsartan, then all sartans came in. So those are B2 products. No really new innovation. Okay. Then came breakthrough innovations. This is only 0.1%. Breakthrough innovations. Now, what is breakthrough innovations? Is conceptually from somebody was using beta blockers to treat hypertension. First time somebody conceived the concept of calcium channel inhibitors. That's something which is a breakthrough innovation. And then there is a disruptive innovation. Treatment of rheumatoid arthritis using a monoclonal antibody. Nobody uses now, I think very few people are taking prednisolone now for treatment of rheumatoid arthritis. Nobody uses that. everybody is now almost shifted to packet drugs that are available. Okay, so that's fresh disruptive technology. So completely orbit changing, which creates a new space. That's what is only about 0.05% of innovation. So, and there is nothing good or bad in this, because if you look at this, you need to spend very few. The cost is less, time is less, certainty is very high that you will succeed in this. Therefore, these are the targets. If you look at this, once in a lifetime, you will come up with something like that. So, what? Nothing good, bad. It will all be decided what is your innovation strategy. And we looked at, you want to be unique, you want to be different, you want to have low cost, you want to have certain advantage, you want to be the best. But you should get the returns of whatever you are planning to do. Moving on. Patents, I have only 2 slides on patents, so I'm not going to, because patents is very highly talked issue, there's a lot of material available. Google University is good on patents. So, what I'm going to tell you is something which Google is not telling you. Okay. So, it is only awarded to innovations. It's a negative right. What I mean by negative right is that nobody, if you get a patent granted, nobody can follow your innovation. But not necessarily you will be able to commercialize your own innovation. Because you might infringe somebody else. So there is, what I am telling you is there is a difference between innovation and infringement. Something which is innovative can be infringed, and something which is infringed can be innovative. Got it? No? Okay. Let me give you an example. Okay? There is a, I come up with a concept that I'm combining, I'm doing a esterification reaction using an alcohol and a carboxylic acid, and I'm using a certain resin, which is a stationary, and then I'm at a high temperature, the carboxylic acid is dissolved in alcohol, and it is passed over that column in a continuous manner. And the resin actually is acidic resin, so it takes away and it facilitates the reaction. And at the bottom, I get the ester. And acid and this thing are completely converted. So the time that is there. So I am using a particular resin which is, let's say, amberlite 420. Okay. Now, there is a prior art which talks about use of amberlite pad resin for preparation of ester, wherein the yield is, let's say, 70%. Okay, the time required, the reaction kinetics are given in that paper or article, which talks about 7 hours it requires in a vessel, 7 hours it takes, and mine is a continuous reaction, so it is instantaneous, right? Still uses amberlite, but I am getting 99% yield. Very pure compound is coming out. So that's surprising property. There is a surprising. There is something which was not anticipated. So there is a. Is it novel? Yes, because nobody has used amber light 420. They talks about amber light generally. I'm coming up with specific amber light 420, which gives a 99% result. Has it been? And it talks about 70. So somebody might think, okay, if I use certain conditions, it might go up to 80, but nobody looks at 99. Look at 99. It's surprising. 99% purity right from the reaction mass. I don't need to purify the compound. I just fill it in the drum and sell it. So there are a lot of surprising properties. So innovation is there, innovative step is there, novelty is there, non-obvious, the industrial utility is there. I get a patent. But if there is a claim which talks about ester using an amber light resin, will I be able to commercialize my patent without licensing that claim? Possibly not. No, I can't, because I infringe. I am using amber light there. So I infringe, but it is innovative, right? Similarly, something which is not infringed. Let's say there is a claim which talks about converting the same reaction in methanol. I am using methanol. Okay. I don't infringe. But is it novel? I need to show surprising property and all these things, and then only. So please don't confuse between infringement and novelty or patentability. These are completely different things. Completely, treat them differently. So the analysis, whether it is patentable, is based on different criteria. Whether it infringes is a different criteria. The same criteria is not applicable. So that's why I'm saying it is a negative right. You will be able to stop others provided you get it. Who you are writing a patent? You are not writing it to the expert. It should enable you what you have done to a person of ordinary skills in the art. We call it procedure. Or you can put that having also, person having or, and it is called as posita also, but normally posita is the term. So you are writing it, and who is this posita? Who is the posita? In case of, take that example of esterification reaction. Who is the posita? He is not the PhD. He is not the master. He is not even the bachelor. He is the person who is your lab attendant, who knows methanol, who knows carboxylic acid, and he can conduct that reaction to get the desired result. What you got? You are writing it for that person. So your specification, your process has to enable that person to produce the result. And it will be different in different cases. So if you are talking about a method of use in your claim, it will go to somebody who is practicing that particular disease, therapy for that disease. So it's different person you are talking about in every claim. And it's the claim what gives you those protection, not the specification. Specification is for enablement, claim is for protection. So what you write in claims has to be enabled in the specification. Something if you don't claim but is there in specification, then so you lost it. So disclose what you claim and claim what you disclose. Okay, and there is it's a separate topic. We can talk for hours and hours on infringement and innovation and on patents. But this is the basics about patent. And now what are different types of patents in pharma especially that we are looking at? There is a compound patent, basic compound. Okay, what is a generic compound? What is a genus? You just, you have a benzene ring and you put R, R1, R2. Have you seen any structures like that? You have a pen? I can use this pen. This is a genus. Because there are a lot of variables that I am talking about. So, one single claim is actually claiming millions of compounds. Because if I go on substituting methyl, ethyl, and then C6H5 and any of the heterocycle group over here, it's a different compound that I am trying to claim. So this is called a genus. Now the moment I draw a specific structure over here, which is, let us say, this becomes a species. Okay. So this is a specific compound. I can claim both. I can claim genus because at the time of genus I don't know which one of that compound is going to help. So I have covered a very broad category of compound. Then as and when my research is getting finalized, I claim a species. Once I have this species, I can claim advanced compound. I can claim a salt of this, hydrochloride salt, bisulfate salt, mesylate, any of the salt, or an ester of this, or an amide of this, something which is prodrug. Okay. Then once I have this compound, further down the line I will find that okay, this is the form 1. It exists in a different polymorphic form, and amorphous is the highest soluble. So amorphous form, or form 1, which is very stable, form 2, form 3, form 4. All the polymorphic forms, or a certain particle size, because that particle size is actually giving me a better dissolution or a solubility. Okay, so all these are advanced compound claims. Then you have process of making this particular compound or its intermediate, or its salt, or polymorphic form, or particle size, milling, or whatever that is. The process of making this, then a formulation of this. The formulation can be, in terms of once a day. It could be twice a day, thrice a day, with a certain excipient, with a certain glided or with a certain coating in a certain particulate dosage form, whatever it is. So there are different formulation claims that I can have. Then how to use this particular compound. Classic case, if you remember, sildenafil, Viagra. First, it came out as treatment of hypertension was the use. Okay, but then suddenly they discovered that it actually inhibits PDE4 enzyme, and therefore it can also be used for erectile dysfunction. So it's a second method of use for wire. The first method of use was hypertension. All the blood pressure drugs, if you see, the secondary indication for them is cardiac insufficiency. Apart from hypertension, you will find cardiac insufficiency as a second medical use. If you look at remdesivir, the first method of use is influenza, the second method of use is COVID-19. Okay, so different method of use of the drug device. Okay, the auto injector, the packaging device, the PFS, everything can be claimed if you are using it for the first time or if you are doing some changes in it. Packaging, how you package a particular product. If it is different, child resistant, like Z-Pack, azithromycin, first when it was introduced, it came and then it came into a child resistant, they called it Z-Pack of 3 for 500 mg and 6 for 250 mg. So they called it a Z-Pack. And they patented it, they claimed it because it was unique, it was different at that point in time. Blood profile. So there are patents which talk about how much should be the Cmax, how much should be the Tmax, how much should be the AUC, how much should be the in vitro release profile in terms of dissolution, how much impurities should be there in a compound, everything virtually. And then supply chain, how you are supplying the product. Okay, so these are called as REMS. Risk evaluation and mitigation system. So how for certain narcotic and certain controlled substances, how their distribution will happen. There are patents on that. How to transport certain medicines. There are patents on that. And those are unique cases. So supply chain. These are some of the areas broadly in which you can claim something. Anything, everything under the sun is patentable, except something which is against the humanity. Who does that? How to kill somebody if you invent a different method? Unfortunately, it won't be patentable. But if you claim it properly, you can still get a patent on that. Probably you have to claim it as a pesticide or something. It is patentable. Everything is patentable provided you have three things: novelty, non-obviousness, or apprenticeship or industrial application. There is a struggle for 3 months or 3 years normally with any patent office, especially with the Indian patent office struggles more. And the cost is quite high.
Finished product containing salt is that cannot be fit. Finished product, suppose it is with different salt, that is not fit. Here the compound is okay, but if it's suppose in a finished formulation, originally it's having a patient with one recipe form or one salt form or without salt form, basic drug. Suppose a formulation is being made with the salt, it is not patented.
It is patentable. India 3D. We are talking from an Indian perspective, that is called as 3D, which is not the only, I would say, the patent office which has some clause like 3D. A salt or ester of existing product is not patentable. Formulation, patentable. No issue with formulation. You can't claim the salt or ester or a polymorphic form of the existing known compound. But it is changing. It is changing. There are 2-3 court cases that have happened which are now taking, I think, by and large, if you look at Indian Patent Act, why in 1975 we adopted or why we discarded the compound patent on pharma products specifically, is because that was the time when our homegrown pharma companies were coming into picture. Earlier, if you see before 1970, it's all Pfizer, it's all, all multinational companies. But now, comes 1980, you will see Ranbaxy's, Sun, Lupin, Alembic, and Cipla, Wockhardt, Glenmark, all these companies started going up, and now India is supplying almost 60% of medicines to the world. 60% of medicines in the world are from India. Finished dosage from India. So we could reach up to this level because we abolished the patent law. We didn't say no compound was not patentable in India till 2005. And that was a great achievement. Because we learned the reverse engineering, we could do everything. And once we understood all these things, once we were ready to face the challenges in the late 1998, that's when under TRIPS we accepted the TRIPS amendment and all those things, WIPO. We went ahead with WIPO, World Intellectual Property Organization. And in 2005, we adopted from 95, retrospectively, from 95, we adopted compound patents even in pharma. But these 20 years gave us a lot of impetus and advantage, so that now we could say that we are the supplier of world medicines. So it's okay, in another 20 years or another 10 years from now, all these 3D and all other things will be removed from the Indian Patent Act. You will have, because if you are planning to become number 3, number 4 in terms of GDP and other things, you can't have the patent law, which is not comparable to the best in the world. You can't expect the other corporations not to have claims in India which they are enjoying that kind of this thing in other geographies. You're party to TRIPS, so we have to be TRIPS compliant. But we have something called as compulsory licensing. So even if we want to follow somebody's new molecule, we can always say that it is a requirement. And in India, you will normally find with any particular disease, if there are about 1 million people affected in the US, in India you will find 4 million people. So it becomes a kind of public necessity or healthcare necessity, and all patent laws can be used for that particular product. And compulsory licensing that we have. So on formulations, yes, you can have as many claims as possible on formulation. Not 3D restricts you to file on the compound per se, if it is a salt or a ester or a polymorphic form of existing known compound. But again, it says that if there is a significant, unless there is a significant advantage in terms of efficacy. So you have a better efficacy. That's what GSK tried to do, right, with this imatinib. Clear. That the other dosage forms you don't have efficacy because it doesn't dissolve. But efficacy is, the Indian Patent Act looks as efficacy as in vitro efficacy. It doesn't look at in vivo efficacy. Otherwise, if you look at polymorphic forms, half of them are not soluble. So, any question so far? So, your innovation strategy is to be very clearly defined when you are looking simultaneously at the patent also. So, your hypothesis has to be right first, then you are looking at what experiments you are going to do. So, doing is very important. Then you need to look at, you are not just curing the symptom, you are in problem solution, you are curing the disease. So you are not just doing a me too innovation, but that incremental innovation also aims towards a resolution of issue rather than just touching it from surface and avoiding the side effects. Yes. Approaches, planning, very critical, because in an institute you will be always short of resources, funds, but there is no shortage of manpower, fortunately. So you have a lot of manpower available in the industry, in the institute. So you can use that. Also, reconsider others' failures. Somebody has failed, don't talk to others. You know why this happened. And what I have found is many researchers are open to discussion. You should ask the right questions in a right tone. Yeah? Okay? This you know. No. So, you should ask the right question in the right manner. That's intelligence. And you will get all the answers why somebody failed. In fact, if you ask somebody a question that you're not able to do it, that's the best thing you can do. It helps that person also to get a lot of dopamine because he's actually thinking he's giving you a lot of solution. Okay, and you get a different solution. Whether to use it or not is up to you. But you at least get a solution. So rather than having meetings, what I think is very important is bad news meetings. What went bad in this week? Talk it freely. So there is no stress and you're throwing open your problem. So I was trying to do this thing, I couldn't do it because of this issue. And somebody may suggest you something, or there are 10 people listening, at least 5 people will give you a solution. So you have 6 solutions, 1 of yours and 5 of others. So, and this is something which we practice. I have started practicing since 2013. I'm practicing this bad news. And it helps, tremendously helps. First, it takes away a lot of stress because something not happening in researcher's mind is stress. So, when you throw open this problem to 10 others, you are getting those 10 others to also be a part of your stress. So you are dividing that stress. You become stress-free. Execution latches. Now execution, what do you mean by execution? Okay, up to planning, 90% of strategies fail because they are not executed properly. What is execution? Execution is day in, day out. Do the same thing. 90% of us are executors. We don't, in smaller way we conceive the vision, the planning, but what we do, day on day basis, we just do things, right? This has to be done, this has to be done. आज ये पीरियड ये है, ये क्लास है, हो गया आज का. That's execution. Okay. Now, if we follow and read a book on execution, there are some really excellent books on execution in terms of management principles, how to execute. But execution is a very simple process. But most importantly, you need to have the buy-in of everybody involved in execution. Then only it is going to happen. You should have those resources when they are available at that point in when you planned it. So you've got to just make sure that everything is available before even starting something. Halfway down the line, you should not realize, oh, that has not come. Something like that should not happen. So execution is very critical, and also very importantly, if you're doing something which is research, I think you have to look at scale up. You have to look at technology transfer, how easily that can happen, and what is the commercial angle for that. Then that probably has some better application and in terms of revenue generation, because what is happening since 2015-16, since the new government has come up, the industry-academia partnership is improved. And if you get into the league of CDRI, or which are government institutes right now, but there are certain universities which are not government universities or institutes, but still they are in demand with the industry. Industry is doing a lot of research. So, one thing is industry coming to you by when you are approaching them, but your just reputation, it takes time. So your strategy has to be like, within 5 or 6 or 10 years. The industry should come to us for this particular area. Can't be that can't be generically for everything, but specifically you need to define that I'm going to create specialization into these areas, which are these important things, and industry should come to us for doing their research in these specific areas. They should not go to any other institute or any other college or university. They should come to us. And then start thinking of differentiation. So that could be one of the strategy. Other strategy could be you create infrastructure and slowly develop the kind of expertise that are required in certain areas. So you become a contract research. It's a fee for service model. It's fixed manpower, it's not project based. So industry can come, or your own students who want to become entrepreneurs can use those facilities as a seed or starting point where lot of different equipments and everything is commonly available. So there are certain common points which the student doesn't have to invest. He can just pay you some rent and can still get the work done. And you will find a lot of that. Many even alumni at some point in time. I would have preferred that if that option would have given to me back in 2016. Somebody would have said, okay, we have an infrastructure, just come and I had to spend 4.5 crores on creating that infrastructure. That money I could have used somewhere else. And there is funding available for that, 10 crores. ATAL. That scheme is called as ATAL Innovation Center. 10 crores of funds are available at a very, something like 2.5–3% plus whatever is the interest rate. So normally would come to around 9%. Collateral free. So you don't have to put any collateral. It's a collateral free funds. 10 crores.
So thank you. Any questions you have?
Most welcome.
So just summarize so please take it. Yes, I would.
Yes, I would like to appreciate the effort taken by Dr. Mandar Kodbule for making us understand this topic. It was a very, actually I want to share, it would be the same feelings with the audience as well, and I'm literally fascinated with the knowledge you have shared today through this topic. I would just like to summarize today's topic. Yes, the contents of today's seminar were importance of knowledge and its use in R&D, innovation and patents. A new term, ligometry, was introduced, wherein we need to actually understand that what we don't know first. We need to quantify what you don't know. We have to make an effort. And a quality planning is very important. We should have a problem-solving approach. Then sir also explained about knowledge management in every college is important. Giving few examples, when we started off in 2014 with the Harvard University, where 100% problems were related to the knowledge management. Then sir also, I can say, motivated us to learn through our mistakes. It's like to early fail and fail cheap was the words of wisdom maybe I can add on. Rather than going on to the end and making mistake and having a crores of loss. Then sir also explained about the prior knowledge of research, identifying the problems through it, coming up with the hypothesis, then designing an experiment depending on the hypothesis, running the analysis or running the data, getting results, concluding and moving way forward. Then research is basically anything that has a problem-solving approach. Then sir also made us understand the scale-up issues, which is the commercialization dynamics. Then in innovations and patents, innovations is something which is not known and doing it for the first time. There are many disruptions examples which sir shared from Mac in 1984 till iPad in 2010. Similarly with Pharma, an example of diabetes was shared. Then another word of wisdom was innovation never stops. The strategy related to this innovation is art of creating superior performance, and planning is just a part of it. Then innovation strategy, wherein we have to know that our idea is unique or different rather than being best. Then planning depends on goals. Then pharma innovation trends were also discussed. wherein in 50s and 60s reducing mortality was important. Then addressing the problem was in late 80s and 90s, reducing the side effects. Then in 2000s, innovation train came up, improving the tolerability, reducing the side effects. Then in 2005, faster recovery, and now working for quality of life improvement. Then sir also made us realize about the pharma expenditure or spends around 70 to 80 billion US dollars in a year for innovation, which is much more than any other field. Sir also made us realize the challenges in pharma innovation, which can make the work time consuming, and there are rate of failures increasing, rising cost of pharma, regulatory incompliance, the targets which we have to meet, innovation capacity which is globally going down by 33% was also made realized. One more thing that sir said, we are all in right now in Google University. Then the other trends were with new chemical entities and having new chemical targets, drug repurposing, improving profiles of the existing drugs, example in oncology, then finding new use of discarded products, new combinational therapy should be tried with the synergistic effect, reducing the dose of the drug which has more side effects, improving the care, pre-operative also and post-operative, and altering the side effects. Sir well explained the dextromethorphan and the quinidine example. Then new initiative with respect to patient compliance, which is reducing the dose frequency, improving abuse potential, having smart devices or delivery devices available, then personalized medications by 3D printing technique and compounding, then also utilizing natural products for the usage, then innovation trends with the emerging therapies, specifically diabetes, oncology, antiviral, monoclonal antibodies, immunological, obesity and pulmonary. Then innovation trend in new chemical entities and dosage forms were discussed. Majorly emerging ones are the novel drug delivery systems, liposomes which are utilized for oncology, matrix techniques, gastro-retentive technology, then reservoir techniques, capsules, which is specifically which a new thing was a stress fit enclosing 1.5 grams of the dose. Then biodegradable microneedles were discussed. So it also made us realize the excipients and the packaging devices are important for the delivery of the drug. So along with API, these are also very important parameters to be looked upon. Then the new emerging areas were discussed, like regulatory, quality, compliance, pharmacovigilance, QBD, technology management, strategic planning, intelligence tracking was explained. Then innovation categories, which were incremental innovation, where can has a potential of 99% of the patents can be filed under this. Then we have Me Too, breakthrough innovations, disruptive innovations. Then the third topic was patents, wherein it is meant to be an awarded to innovations. There are negative rights for patents. Then the three parameters of patents with novelty, inventive step and commercialization applications was also explained. Then claims is for protection and should be enabled in specifications was well put up. Then infringement and patentability are two different parameters were also discussed. There are different types of pharma patents which sir included in his presentation, with basic compound, advanced compounds, process, formulation, method of use, device, packaging, drug profile, supply chain. And then the last content was creating innovation strategy, where design of experiments, problem solving for curing a particular disease, approaches and planning, considering the resource and funds available, then reconsidering the other failures, then avoiding execution latches, and successful IP and technology transfer record was also included. Another word of wisdom was something is not happening is researchers' stress, which should be noted. So sir, it was a very elaborative session, and thank you for making here, we are actually fortunate to have you here and making yourself available, accepting our invitation at a very short notice, so we appreciate that as well. And now the session is open for question and answers.
It was an eye-opener for us because like we have a direction now, like how should we focus on research and in what aspects we have to look into. And basically one question I had in my mind, how to look out for unmet needs. Because reading journals may give you some idea, but to know what is needed currently, what is the need of the hour, like what are, so how do we find those unmet needs, and so that we find the research problem?
There are various forums available online, within Google University also. So, the other way of looking at it is go to the WHO World Health Organization sites, look at the demographic issues that are put up in the WHO website. They talk a lot about pandemics, endemics, the endocrine diseases, the CNS. Every country has some kind of health ministry. So if you go to our health ministry website also, you will come to know how much population. That's one way of doing it. You can go to MHRA website, NHS in UK, or kind of NSA on the US. You'll get a lot of demographic details. Also, you will find, if you go to regularly visit the USFDA website, you will find new drugs that are coming up, or the new targets that are being addressed. Every month, I think some or the other, group like Evaluate or City or all these, they will come up with what's next in pharma. What are the issues in pharma? Unfortunately, what happens is we don't put those as alerts. So, if you have Google, if you have a Gmail or you have any kind of this thing, go to Google, put alerts on the area in which you want what is happening. You will get a mail. In the morning, might be you will get 400, 500 mails. You have to scan through that to know what you are looking for because something which is opportunity for you may not be an opportunity for somebody else. What is unmet need? So for identifying unmet need, everybody's criteria is different. Okay, and it will also depend upon your own capability. So, what there is an unmet need about long-acting injection for treatment of schizophrenia, for example. I know that, but is it something which I want to work on? Do I have an expertise? No, absolutely not. I'm not. None in my team is a particle engineer expert. None in my team understand how these PLGA, which is polylactide glycolic acid, these polymers are going to be biodegraded inside the body. My team doesn't understand, but my team understands something which is in oral, something which is in normal injectable. Put us any problem, we'll resolve it. That confident. But something which is this, I am not work. It's an unmet need. Do I want to work on it? No, absolutely not. Narcotics is an unmet need. There is a lot much can be done in narcotic products. Do I want to do it? No. I don't want to do it. Oncologicals. Huge potential, right? It's the biggest area. Strategically, we have decided we are not going to work on oncologicals. So, when you are looking at your unmet need, you should, what you want to do, you should also know what I don't want to do. What is my trade-off? So, you should know what is your trade-off when you are doing research. That I want to do this, but definitely I am not going to do this. Even if it's a billions and trillion-dollar opportunity, I am not going to use it. Because that's not my expertise. There are issues that are involved over there. There may be long-term, there may be very cost-intensive research, for which I don't have the bandwidth to attempt those. So I'm not going to touch that. Therefore, what is an unmet need or opportunity for any particular individual or organization or an institute is a subset of what is your capability, what is your intelligence tracking ability, what you understand. For example, I was looking at there is one product which was approved, which was in phase 2, in fact, itraconazole 65 milligram product. Okay. 65 milligram equivalent to 100 milligram of Swaranox. The brand name was Tolsura. We identified that product. Looks to be good, right? Because itraconazole $75 million market, at least 10% market share with a better pricing, it can reach to about $30-40 million. Let me be the first to capture that. So we developed the product, kept it ready. The product got approved. Unfortunately, it didn't even become a $1 million product. There is a huge potential for the product, but it then so my assessment, competitive assessment that it will have some potential was wrong. So I had to, and I had spent money in developing that product, maybe about 30-40 lakhs. Because I don't have a spray dryer, it requires a spray dryer. Itraconazole as an API, the polymer, everything. It's costly. Costed me 40 lakhs. What to do? That's it. So you should be also ready to course correct at the right point in time. If it is not working, forget it. Whatever. So if I would have came to know about it when I had spent 5 lakhs of rupees onto that, I would have been more happy, right? But it has told me something. The learning is, if it is a anti-infective kind of treatment, don't take anything as an assumption. Because simultaneously, at that same point in time, a new drug got approved, which is isoconazole. I failed to track that isoconazole is getting launched, so whether this 65 mg itraconazole will be of any value or not. So unmet need is not unidirectional. You have to have, what are the other products in the same segment, what is happening on them, when are they going to be launched, what is the intent? Is it better or is it has lesser side effects or better profile? Everything matters. But to begin with, the best thing is put Google alerts on the areas that you are interested in. You will every day, by default, Google will send you fantastic mails in which you just have to see what is happening. If you think that is useful, use it. If you don't think it is useless, delete the mail.
So with due respect, but as we are falling short of time and running behind the schedule, with sir's permission, I request the participants to put up their questions to Dr. Anita Aire Madam, and she will forward it to sir. And
you have my email address also
yes.
there, so feel free to write, call.
Thank you, sir. I request Principal Madam to felicitate sir and congratulate sir for a very wonderful event.
Thank you.
Thank you so much. It was a wonderful. Correctly understood the need of the hour and you touched upon the questions. So,